So, good evening, everyone. This is your fifth week session with me. And in this session, we'll be looking into the zinc finger nuclease technology, its basics, and how to design the zinc finger nucleases for genome editing. And we'll also look into the applications of zinc finger nucleases in different fields. Then we'll practice MCQs. We'll also look into the current literature, and I'll take your doubts. So, before moving further, we must understand what is central dogma. So, central dogma of molecular biology, it is a theory which states that the information flows in only one direction. That means the information pass on from DNA to RNA and from RNA to proteins. So, this is the unidirectional flow of information, and it says that from information will not pass from protein to DNA. So, this one-dimensional or one-directional flow of information is known as central dogma. This is the fundamental theory, and it was developed by Francis Crick in 1958, and his version was a bit different and it was more global. It included that information does not flow from proteins towards the nucleic acids. But later on, different scientists have discovered that there are several exceptions to this central dogma theory. And for example, prions. So prions are one of the example in which they are the infectious proteins. So they do not require DNA. They are infectious proteins which replicate without going through DNA to RNA intermediates. So these are responsible for the rare but devastating neurological disease, which is the Creutzfeldt-Jakob disease, and which is also lethal. So there are these are some of the exceptions that DNA is not the only component which is required for the flow of information, but in some prions, protein can also act as a main source for the flow of information. And different viruses have also shown the process of reverse transcription, in which they convert their RNA to DNA, and that DNA is known as cDNA because the processed RNA lack introns. So the cDNA will also have only the coding region. So this type of flow is also reverse flow. So this also violates the principles of central dogma. Okay, so I'll just quickly again go through the concept. What I mentioned is, so the central dogma, it is a flow of information in one direction. So according to Francis Crick, he mentioned that information flows from DNA to RNA and then to proteins. But different scientists have identified that this is not true. There are some exceptions, and one such exception is prions. So these are the organisms, or we can say these are just the infectious proteins. And for their replication of for their division, they do not require DNA or RNA. So this violates the principle. This does not follow the principle that DNA is required to flow the information. And another example is of the viruses, which can convert the RNA to DNA. As soon as they enter the host, they replicate their RNA and integrate with the host, or either convert their RNA into the cDNA. So this is all about the central dogma. Now we'll look into the protein structure and its building blocks. So as in case of DNA and RNA, we know that the building blocks were the nucleotides. Similarly, in protein, the building blocks are the amino acids, and this is the basic structure of amino acids. So there are total 20 main amino acids, and some other amino acids are also there. So this is the basic structure. Each amino acid will have a central carbon, which is known as alpha carbon, which is linked to one hydrogen and one R group. So this R group is alkyl group, which will vary among different types of amino acids. Its N-terminal end, or its 5' end, includes the amino group, which is the NH2 group, and C-terminal end, or the 3' end, includes the COOH group, which is the carboxyl group. So in each amino acid, you'll see there will be a amino group, a carboxyl group, a hydrogen, and a side chain. And if we compare different amino acids, then they will vary only in their side chains. And on the basis of side chain, these amino acids are categorized into different types. They could be acidic, they could be basic, they could be polar, non-polar. So whenever two amino acids join to one another, so there is loss of water molecule and the bond which is formed between two different amino acids. So this type of bond is known as peptide bond, which is formed between the carboxyl group of one of the amino acid and amino group of the another amino acid. So there is loss of H2O molecule and there is a bond formation which is CONH. So this CONH bond is no peptide bond, and with the help of peptide bonds, different amino acids join to one another and hence they form a protein. So as you can see, this is a long protein chain in which These small circles represent a single amino acid. So these are series of amino acids which are linked to one another with the help of peptide bonds. And for example, if we look at the C terminus, so N terminus and C terminus mainly shows that the protein starts with the means one of the amino acid which is present at the terminal, its NH2 group is exposed from its 5' end. That's why that terminal is known as N terminus, where the amino group is exposed. C terminal is the one which is the 3' side of the protein in which the C terminal group is exposed, which is our carboxyl group. So that is how the proteins are, the directionality of protein is referred to as N terminus and C terminus. So for example, these four circles, they represent these amino acids, leucine, phenylalanine, then cysteine, and then instead of serine, they mentioned set. So there are some additional amino acids which are also formed during the respiratory and other enzymatic processes in our body. So this represents the linear chain of amino acid, and bond between each of the amino acid is peptide bond. Yeah, and this R group. So in case of phenylalanine, this is CH2 and a phenyl, a benzene ring is present. So each amino acid will vary in its side chain, which I mentioned. Okay, so this protein structure is mainly divided into four types. So first one is primary structure, in which we look at the linear chain or linear sequence of amino acids which are present in the protein. So primary sequence will be like, for example, if there are alphabets. So if we talk about the English literature, English language, then the alphabets are like our primary structure of a protein. Then if we talk about the secondary structure, then in this secondary structure, we look at the how these amino acids are linked to each other with the help of hydrogen bonding. And these are like small structures which they form. So these are like smaller substructures such as alpha helices, beta sheets. If beta sheets are arranged in parallel, then they are parallel beta sheets. If they are arranged anti-parallel to one another, they are anti-parallel beta sheets. So in secondary structure, we mainly look at the hydrogen bonding of these peptide backbones, which cause the amino acids to fold into a repeating pattern. So these repeating patterns will be observed in the entire protein structure. So that is known as secondary structure. So in terms of English literature, if we want to see, so the words are like our secondary structure. So this shows that how, as we expand from primary to secondary to tertiary, how we increase the knowledge of the structure. So previously we only had the alphabets, we only had the amino acids which are present in the protein. Now we also have their bonding, the hydrogen bonding which they make. So that gives us the words, that how these alphabets are involved in making the words. Now if we talk about the tertiary structure, then it is the three-dimensional folding pattern. So if these alpha helices, beta sheets, if they further fold, then the structure which is like a functional structure. So there are some side chain interactions. So not just hydrogen bonding, but if that R group which we have seen previously, if they also interact with one another, then the structure which is formed, that is referred to as a tertiary structure, which is further folded. So in terms of English, we'll say sentences. So how words are used to make sentences. So this is giving us more precise detail of a protein. Then if we look at a quaternary structure, so this is the structure if more than one chain is present in a protein. So then how the different chains present in the protein are interacting with one another. So that is referred to as the quaternary protein structure. For example, in case of English language, if we talk about the paragraphs, that how these sentences are divided into different paragraphs. So this is how the protein structure is categorized, starting with the primary structure to the quaternary structure. So quaternary structure is the one which will give you the complete picture of the protein with all the details, how it is folded, which are the bonds it makes, what are the hydrogen bonds, and what are the other side chain interactions which it is making. So this is about the protein structure. Now we'll look in detail into the secondary structure, which is the most important one, because in tertiary structure also these secondary structure components keep on repeating. So proteins are the linear polypeptide chains which fold into the repeating structural units. So you'll see that in a protein there could be multiple such structures. Multiple alpha helices can be present, multiple beta sheets can be present. So alpha helix structure was given by Pauling and Corey in 1951. So in this case, there is a hydrogen bonding between the NH and the CO group. So the hydrogen bond present between double bond O and NH, H of the NH group, this is known as the hydrogen bond. So in all the this folded in all the amino acids, we'll see that how many hydrogen bonds are present and how the protein is folded. So in this case, the residues which make this hydrogen bond is the first residue and I plus 4 residue. So for example, I is my first residue, then the bond will be formed between the fifth residue, that is 1 plus 4, 5. And there are multiple type of alpha helices on the basis of which we will see that where the bond is present. So in the next slide, we'll see that. Then the basic type of alpha helix is this helix in which the bond is between the I and I plus 4th residue. So in this case, this one single turn, there are 3.6 residues which are present, and the pitch, or you can say the height of this particular turn is 5.4 angstrom. And this is also known as the pitch of the alpha helix. And the shape is the rod shape. So its shape appear like a rod, or you can say it's like a spiral shape, and it is stabilized by these hydrogen bonds. So these hydrogen bonds are responsible for the stability of this rod-like structure. Then secondary structure is the beta sheets. So these are the beta sheets, and these are composed of linear beta strands which are joined by the hydrogen bonds. So here also the linear chain of amino acid is running, here also linear chain is running, and then there are bonds present between these two linear chains. So with this they make the beta sheet, and these individual chains, these are known as beta strands. And though the two beta strands, when they join together, they make a sheet, beta sheet. So in this case, these strands may be far apart in sequence. So it doesn't mean that it is, if it is beginning with 1, 2, 3, 4, 5, 6, 7, then it is not always that here you will find the eighth residue. It is possible that in between there are some turns, loops, or some alpha helix might come, and then again this strand comes. So it says that the order is not linear. There could be multiple other amino acid residues which can be present in between the two beta strands, which are joining to one another to make a beta sheet. So there are two types of beta sheets, or you can say they can be classified into multiple types depending on how the beta strands are oriented. So it is parallel if all the strands are pointing from N to C direction. So for example, as if we say here, so it is beginning with the N terminal and here it's C terminal. So in this case, both are having different directions. So this type of strand will be my anti-parallel strand. But if it begins with NH and the other strand is also starting from NH, then that type of strand will be my parallel strand. So in which the direction will be same for both the strands. The direction of the side chain will be exactly identical. Okay, so this one is the anti-parallel beta strand, and there are possibilities where the mixed beta sheets are also observed, in which the approximately, if we talk about all the beta sheets, then nearly 20% are like mixed. So in between, it starts with anti-parallel and then it somehow becomes parallel. So they are referred to as the mixed beta sheets. Okay, so now this is further representation of both the alpha helix and beta sheets. So alpha helix can also be referred to, means there are some other names. So first is the alpha helix, which is the main alpha helix structure, which is mainly observed, which is also referred to as 3.613 helix. But there are some other helices such as 310 helix, so which is also observed in some of the proteins. So in 310 helix, you will see that the bond which is made, it is between the I and I plus 3 residue. So in the normal alpha helix, the hydrogen bond which was present, it was present between the first amino acid and the fifth amino acid. Whereas in case of 310 helix, the bond will be between first amino acid and the fourth amino acid. So in this way, this particular helix will be shorter. So the pitch which we have seen 5.4, so it will decrease because the bond is near to each other, and the bond is made between the carboxyl oxygen of the carboxyl group and the hydrogen of the amino group of the other amino acid. And there is also pi helix, which are also not that much common, but they are observed in some of the organisms. So in pi helix, there is the amino acid residues which are involved in one single turn are 4.4 amino acids, and the bond is between I and I plus 5. So that means first amino acid. So carboxyl group of my first amino acid will make bond with the hydrogen of the uh, sixth residue. So this is how different helices will vary in their shape depending on that how long they are making the hydrogen bond. Means after leaving how many residues they are making the hydrogen bond. And this is the representation of anti-parallel beta sheets. So as you can see in both of them, the direction from where they are starting is different. So it is beginning with the C terminal, which is C double bond O, and it is beginning with the N terminal, which is NH. So that's why the direction is different, due to which they are referred to as anti-parallel beta sheets, and their R groups are facing towards each other. So this is also the reason that they are anti-parallel beta sheets, and in most of the cases anti-parallel beta sheets are unstable due to the side chain, because if the side chain is a bulky group, for example, in case of phenylalanine, so it has the methyl group along with the benzene ring. So if two of the side chain will face toward each other, and if they are bulkier groups, then they will repel each other. So the strength of hydrogen bond will not be sufficient to compensate that interaction. So in that case, the anti-parallel beta sheets are generally unstable. So to make it more stable, uh, only those proteins will favor the anti-parallel beta sheet in which the R side chain of both of them, either one of them should be the light group and other could be bulky. But yeah, but if we compare it with the parallel, then they are comparatively uh, less stable. And in case of parallel beta sheets, both of them starts with the same direction. So in both of them, they are beginning with the N terminal and they're ending with the C terminal. So this type of beta sheet is referred to as parallel beta sheets. And in this case, the R groups are away from each other. So these side chains, they're away from each other. So they will not make the steric clashes, due to which the parallel beta sheets are much favored as compared to the anti-parallel beta sheets. Now we will see that how these protein interactions, they drive different cellular events. So as we know, in our body, there are multiple proteins, so they interact with one another to bring about the functionality. So they are dynamic biomolecules. That means they keep on moving, they keep on changing, and they not only interact with the small molecules, but with other macromolecules also. So proteins not just interact with other proteins, it also interacts with DNA, RNA. So if you remember that in case of replication, we see the DNA polymerases, different enzymes. So enzymes are also the class of proteins. So in that case, we have seen that how the proteins are interacting with the DNA. Similarly, in case of RNA also, we have seen RNA polymerases which are interacting with the RNA. So they come under the DNA protein interaction or RNA protein interactions. And these interactions are crucial for the regulation of gene expression. Because we in the previous session, we have seen that how the Cree recombinases, how they decide that when to turn off and in the presence of which of the chemical compound we should turn on the genes and when the genes will be turned off. So all that comes under the regulation of gene expression. So this is also governed by the protein interactions. Proteins bind to the DNA and then they do not allow the other uh, enzymes to bind to initiate the expression. Similarly, signal transduction, so the communication between the cellular components. So in that case also proteins are involved, because our cells generally have some of the receptors where the proteins bind and then they enter into the cell and then they further starts a signaling process. They send the signal to the nucleus, and then some of the functionalities shown, nucleus will uh, regulate the expression. Some genes will get expressed based on the signal, that what kind of signal is coming. Then enzymatic activity regulation. So enzymes are type of proteins. They also make sure whenever to activate a particular cycle or whenever to inhibit that particular process. Similarly, structural organization. So processes such as chromatin remodeling, cytoskeletal assembly, these are also performed by different proteins. They decide, for example, the histone proteins, which we have seen that how the histones, how the DNA wrap around the histone proteins. So if that wrapping around the histone protein is very tight, then the DNA will enter into the heterochromatin structure where it will not be able to express. Whereas if the genes are need to be actively expressing, then in that case, histones will further on get deacetylated and they allow the particular region to get converted into euchromatin where the genes can easily express. So that kind of shift from heterochromatin to euchromatin structure is come under chromatin remodeling, in which it decides that whenever the what kind of genes need to be expressed, because we know that in our DNA it is a long DNA in which multiple genes are present and not all the genes get expressed at one particular time. So some genes remain are silent, whereas other genes keep on expressing. So that all the process, that fate, that which genes get expressed is decided by this chromatin remodeling. And then in the case of cytoskeletal assembly also, the structures, we see that how different tissues are different from each other, different cells are different. So this differences comes from the cytoskeletal assembly, which tissues will give rise to what kind of structure. So proteins guide them. Similarly, there are different types of interactions, protein DNA interactions, which we have seen, transcription factors, nucleases, polymerases. So they shows protein DNA interactions, whereas protein RNA interactions. So in case of RNA binding proteins, the proteins which bind to RNA and then they initiate the process of transcription or some other processes. Similarly, splicing factors. So these are also the proteins which bind to RNA and they remove the intronic region and join the exons. So that process is known as splicing. Similarly, ribosomal proteins. So ribosomal proteins also interact with the RNA because in the ribosome we have different types of rRNAs which are present, which are responsible for the formation of the ribosome. And if we talk about the protein-protein interactions, then in the case of ribosomes, we have two different subunits and different proteins come to form the ribosome. In case of replisomes, so replisomes are the proteins which are involved during the replication process. So when DNA is converting into another, it's making the copy of DNA. In that case, different proteins come such as helicases, topoisomerases, DNA polymerases, gyrases, and many other enzymes. So that complex is known as replisome, in which different proteins complex together, and then transcription complexes. So this shows that how different proteins come together and perform some functions, so that come under protein-protein interactions. So main key concept from here is that protein interactions, they act as a molecular switches or regulators of different genetic and cellular processes. Yeah, so this is the example of protein DNA interaction in which different proteins such as helicases, topoisomerases, SSB, which is single strand binding protein. So they are interacting with the DNA and helping the DNA to replicate. Okay, now let's look at the function of DNA binding protein. So we have already discussed it a lot, but we'll quickly go through it. So in the case of transcription, the binding to the promoter enhancer region to regulate the initiation. So to initiate transcription, because transcription is the conversion of DNA to RNA. So DNA act one of the strand of DNA, which is from the 3' to 5' direction, which is also known as the nonsense strand. So that converts to the mRNA. So in that case, there are some proteins which are the promoters which bind to these regions from where they regulate the initiation of transcription. So in case of replication also, we have seen that the region from where the replication begins, that is AT rich, because those regions are easy to unwind. So in that case, similarly in case of transcription also, there are some Tata boxes. So these are referred to as the enhancer regions where the promoter will bind and it will initiate the transcription process. So the protein which is binding the promoter, that is the protein. So this interaction of promoter to the region is the protein DNA interaction. Now, if we talk about packaging, then the DNA is wrapped around histone proteins and it convert into the chromatin structure. So in this case, the DNA binding proteins are responsible for this packaging of the DNA. Then if we talk about rearrangement, then in the case of homologous recombination or site-specific recombinases which are involved in the recombination process. So it also, these proteins also help to rearrange the fragments of DNA. So due to which it can now bring about the um, diversity in the same protein. So in the case of human, we have the VDJ recombination. So we have different other specific enzymes which are responsible for this recombination processes, and it is generally important in immune system. Then in replication also the proteins are involved, such as DNA polymerases, helicases, primases. And in repair also we have seen that how the different proteins which bind to DNA, such as endonucleases, ligases, which are involved in the DNA repair. So this shows you that how the proteins are playing important role in the functionality by binding to the DNA. Yeah, and this is also the example in which I have shown that how the protein DNA interaction takes place. And this is the chemical structure of those means proteins. that how the side chain groups are present. Similarly, in cross-linking also, the proteins are involved. So DNA protein cross-linking is also observed. Similarly, in case of in modulation also the proteins are involved. They modulate whether to start the transcription or whether to turn off the transcription in the presence of different signals. That signals could be some chemical compounds or it could be light or anything. Yeah, and this shows you that how the double-stranded DNA, when it is wrapped around the histone proteins, then it get folded into the smaller structures, which is around 30 nanometer fiber, and then this further get compactly folded into the chromatids, and then ultimately we see the chromosome. Yeah, so this chromosome is the DNA which is means compactly wrapped around the histone proteins. Yeah, now we'll look into the structural analysis of protein DNA complexes because the topic for today is the zinc finger nucleases. So before moving further, because these zinc finger nucleases, they bind to DNA. So they are like DNA binding proteins. So to understand them, we are making the background. We are starting with our basics. So structural analysis of these protein DNA complexes. So around 240 complexes were studied by Lascombe and different other scientists in 2000, and they have identified that there are 8 groups which could be classified into 54 families of DNA binding motifs. So what are motifs? These are the smaller structures you can say which continuously get repeated in the proteins, so these are known as motifs. Motifs are small, and if we compare it with the domain, so domain is a bigger structure and motif is a smaller structure. And because these keep on repeating, that's why they are referred to as motifs. So for example, in this case, as you can see, this similar structure, which is this loop-like structure, it is keep on repeating 3 times. So that's why this is referred to as motif, which is the smallest structure, which you could see repeating multiple times. So that is referred to as a motif. And there are 8 groups in which these 240 complexes were classified. So these 8 groups are helix turn helix. So this is the helix turn helix. These are the motifs. So one helix and then there is a turn and then it is means joined to another helix. This kind of motif is referred to as helix turn helix. And okay, so in these the two helices are present. So the plural of helix is helices. So 2 helices are present. One is responsible for stabilizing and another one is the recognition helix. So in this motif, one of the helix, it will identify the region in the DNA. So now these motifs are present in the proteins. And now we are talking about protein DNA complexes. So this particular motif, one of the helix is responsible to stabilize this structure and another one is the recognition helix, which will recognize the DNA strand and it will bind to our DNA strand, and then it will bring about the protein DNA interaction. And generally it binds to the major groove of DNA, and one of the example of this type of motif is lambda Faj C2. So in this particular lambda Faj C2, the motif which is observed is this HTH motif. Another is zinc coordinating, which is uh, so this one is the zinc finger motif in which it coordinates with the zinc ion, Zn2 positive ion, which stabilize this motif. It includes C2H2 zinc fingers, zinc ribbon and ring fingers. So in this case, C2H2 is not carbon hydrogen, it is 2 cysteines and 2 histidines. So these cysteine and histidine, these are the amino acids, and as we are showing a protein, and these are the motifs which are smallest. So here we are only representing the smaller part of a bigger protein. So this particular motif, this is our zinc finger motif. In this case, we have 2 cysteines, 2 histidines. They coordinate with the zinc ion, Zn2 positive in between, which is not shown here. So that's why they are known as zinc coordinating DNA binding motifs. And C2H2 zinc finger name is also given to them because they have 2 cysteines and 2 histidines, which are responsible for that interaction with the zinc ion. Then another type is zipper type. Okay, so this is like a leucine zipper motif in which leucine is uh, coordinating between the 2 zippers, and this is the zipper. So this structure appears like a zipper. That's why its name is the leucine zipper. Then alpha helix motifs in which multiple alpha helices are present. They directly contact with the DNA grooves, and in this case, these are not helix turn helix. So for example, helix loop helix is the alpha helix motif, in which there is no turn, but there is loop which is present, and different helices are joined to one another, and these helices directly contact with the DNA grooves. Then beta sheet motifs. So these are rare, in which beta strands insert into the DNA grooves. So as the name suggests, beta sheet, that means beta sheets will be present. Then there is beta hairpin or ribbon. So beta strands linked by the tight loops slash ribbons. And example is MET-J repressor. So this is the example of beta hairpin, because the structure looks like a hairpin-like structure. So in this case, these are the anti-parallel beta strands, and there is a turn which connects two beta strands to each other. So this is known as beta hairpin motif. Then enzyme linked motifs. So these are the catalytic proteins that bind the DNA, such as restriction enzymes, polymerases. And then there are some other motifs which are unclassified structures, which could not be classified, but still they are present, and study is going on to them. So some other beta sheet motifs are Greek key, in which there are four beta sheets which are present. So there are this first and second, so they are anti-parallel to each other, and further means there are series of beta strands which are present, which are anti-parallel to each other, and they are joined to one another by these turns. So these are the turns. These are some stretch of amino acids which are joining these beta strands to one another. So starting from here, and then it goes to 2, 3, and 4. This is towards the C terminal, and this is our N terminal, and this is how the structure will appear. So its structure appears like a Greek key, hence the name is given as Greek key motif. Similarly, it forms the structure of a barrel in which multiple beta sheets are running anti-parallel to one another. So this is referred to as beta barrel. This is a complex beta sheet structure, and beta barrels are commonly found in proteins that transport ions across cell membranes. So for these proteins which are involved in the transportation, we'll see beta barrel kind of a motif which is present. Yeah, so these are some of the examples of the DNA binding motifs which are studied, which are well known. And then we'll talk about the FOC1 nuclease. So this is originated from the Flavobacterium ochinocoides. So as the name, so this is how the naming is done. F is taken from the Flavobacterium and oak is taken from its species name. And then one specifies the serial number from whichever lab it has been designed, and the action is the nuclease. So they degrade the nucleic acids. That's why they come under the nucleases. Nuclease is the enzyme category which degrades the nucleotides, and this is the type 2s bipartite restriction enzymes. So when we looked into the restriction enzymes, we have talked about type 1, type 2, type 3. So similarly, it comes under type 2s bipartite restriction enzymes because it recognizes a specific asymmetric sequence. So when we talk about the exonucleases, endonucleases, so nucleases, we only say that these are the enzymes which are responsible for degrading the nucleotides. But when we say restriction enzyme, then the one of the major property of restriction enzyme is that they have some recognition sequence which they recognize and then they cut. So in this case, because it is type 2s, so the recognition sequence and the site of cut is near to each other. It's close. So it recognizes a specific asymmetric sequence which is GGATG. So 5 prime to 3 prime. So whenever we'll read a 5 prime strand, then it will be GGATG. And this direction is important because if you search it in the 3 prime to 5 prime strand, you will not search this sequence. So in 3 prime strand, it will be CCTAC because both of the DNA strands are complementary to each other. So if you read the DNA strand from 5 prime to 3 prime, the sequence is GGATG. So this particular sequence will be recognized by FOC1 nuclease. FOC1 nuclease will bind to this region, but where it will cleave? So it cleaves 9 base pairs downstream in the 5 prime strand. So in this strand, it will cleave 9 nucleotides downstairs after the recognition sequence. And in the 3 prime strand, it will cut 13 base pair downstream. So not just 9, but additionally 4 nucleotides are also included. So after 13 base pair, there will be the cleavage. So it will bring about double strand break in the DNA strand. That is the main work of FOC1 nuclease. And this is the recognition sequence: 9 base pair downstream in the 5' strand, 13 base pair downstream in the 3' strand. Molecular weight of POC1 nuclease is 65.4 kilodaltons, and it is composed of 587 amino acids. So this is the number of amino acids present in the POC1 nuclease protein. And if we talk about its structure, then there is N-terminal domain and a C-terminal domain. So N-terminal domain, so this is the whole protein. This is whole POC1 nuclease. Towards its N-terminal region, from where we start the protein, from where we read the protein sequence. So towards its N-terminal region, there are 3 subdomains. So subdomains means smaller domains which are present. So D1, D2, D3. So in this case, these are the zinc finger domains. These domains can be some other domains also. And why these domains are important? Because these domains are responsible to bind to the DNA. And these are responsible for DNA recognition. Then there is a C-terminal domain, which is this one, which is also known as the nucleus domain, responsible for the DNA cleavage. So in each recognition or in each restriction enzyme, you'll see there will be at least 2 components. One component which is responsible for identifying the recognition site and binding to it, and another component is responsible for the cleavage, which will make the cut. So in this case also, this is how we divide it. So there are 3 subdomains and one cleavage domain or C-terminal domain, which is responsible for the cleavage. Now, what is the mechanism? How it does this its activity? So it binds as a monomer, which is inactive. It requires dimerization. So alpha 4 and alpha 5 helices and loops P1 and P2, they mediate a interface, and active complex cleaves the DNA phosphodiester bonds, because we know that the sugar phosphates, that is the backbone of our DNA, sugar phosphates are linked to one another with the help of phosphodiester bonds. So whenever you want to break the double-stranded helix of DNA, you need to break these phosphodiester bonds, and for breaking them, nuclease activity is required. So these POC1 nucleases, if there is only the monomer is present. Monomer means single chain. So this is the monomeric unit. It cannot function, it is inactive. So it requires dimeric unit. That means it requires 2 chains of the POC1 nuclease to bring about this nuclease activity. And these are the helices which are involved. So in later slides, we'll see that how the secondary, or you can see how the tertiary structure of this particular POC1 nuclease appears and how it interacts. But for now, just keep this thing in mind that there are 2 monomeric units. So now there are 2 separate monomeric units. So now whenever they bind to DNA, they will make a dimeric unit. And then they will recognize the DNA with the help of these 3 subdomains on both the sides. And POC1, this C-terminal domain, will bring about the cleavage. And in the 5' side, the cleavage will be after the 9th nucleotide position after this recognition sequence, and in the case of 3' strand, it will be at the 13 base pair downstream. So this, whenever we read towards the forward direction, this is known as downstream, and if we read it in the backward direction, that will be upstream. So in case of DNA, this is how we name it. So 13 base pair downstream, the cut is made in the 3' strand. Now the application of POC1 nuclease is that it is useful for cloning. So how cloning? Because it can cut the DNA, and in that we can insert our DNA of interest or gene of interest. So it can be used for cloning. Then cleavage mapping. So we can also map that where the cleavage has been done. And then gene manipulations. So with the help of cut, we can remove some fragment from the DNA. So we can manipulate the gene. We can insert, we can delete, we can invert. So all these things can be done. Okay, now this is how the actual structure of POC1 nuclease appear. There are multiple alpha helices which are present, and then there are some beta sheets also. So these colors, these represents the subdomains D1, D2, and D3. And then there is a cleavage domain which is shown with these blue structures. So these rod-like structures are the helices, and ribbon-like structures will be the beta sheets, which are not visible clearly, but this particular arrow-like structure, which is like a ribbon, this is our beta sheet. So if we identify the secondary structure in this particular secondary structure of this protein, then this is how the secondary structure is composed. These are the alpha helices which are present in each of the subdomains. So total, as we can see, alpha 8. That means there are 8 alpha helices in each subdomain and 3 beta sheets. Similarly, in this case also, we have uh, in the another subdomain, we have only till alpha 5. That means just 5 alpha helices and 5 beta sheets. And in case of this white one, which is the subdomain 3, in this case we have alpha 1 to alpha 3. That means 3 alpha helices and 3 beta sheets, beta 1, beta 2, and beta 3. And then in the case of cleavage domain or C-terminal domain, we have nearly alpha till alpha 6. So we have 6 alpha helices and 6 beta sheets. So this is how you can read the structure of the entire protein. But this is just the secondary structure. This doesn't mean that alpha 1 is close to alpha 2. In the tertiary level, they can be organized in such a way that alpha 1 can interact with alpha 6 or maybe alpha 8. So this is at the level of secondary structure where we are simply seeing that what are the components which are present, how many alpha helices, how many beta sheets. Yeah, this shows how the dimer interface is made. So dimeric interface means this fork one and this fork one, these are two monomeric units. So how they interact with each other? So they interact with each other by these this alpha 4 and alpha 5. So these blue one which are present in the C terminal domain. So in this case, from here they will interact. So from this fork plus region which is shown here, this is the C terminal domain. So from the C terminal domain, alpha 4 and alpha 5 helices interacts with the alpha 4 and alpha 5 helices of the another fork 1 protein C terminal domain. The interaction is made between them. So this is the another representation. So for example, these are the three subdomains and this is my C terminal domain which is responsible for cleavage. So this here it shows that fork 1 binds to the first site. So in one of the strand it has bound. Now it is reversible, its structure will convert into this form. So this is how it is oriented as soon as it binds. Now fork 1 binds to the second site. Now similarly, second fork 1 will come, it will bind to the second site. Second site means in the second strand, which is the 3' strand, it will bind. Now both of them will interact with each other with the help of their C-terminal domain. So this is their C-terminal domain alpha helices, and they will may interact with each other and bring about the cleavage. Yeah, so this is how the cleavage is shown. So in both the sides, cleavage will be done. In the upper strand, cleavage will be after 9 base pairs, and in the bottom, it will be after 13 base pairs. This is how double strand break is brought about by fork 1 nucleases. So I hope it's clear till now. Okay, now there are some reviews. If you're interested, you can go through this, which also gives you more details that how these endonucleases work. So this particular review talk about that how the endonuclease are the new tools to edit the mouse genome, because mouse is the closest model which resembles the human. So whenever if you want to treat, we want to design some therapy for humans, so we first test it on the mouse. So endonucleases are the better tools, and fork1 also comes under endonucleases, specifically restriction endonucleases or restriction enzymes. And this shows that how the fork1 binds to the double-stranded DNA, and these are the three subdomains. So in this case, they are the zinc finger domains. And then they bring about the cleavage in which there is a double strand break. Now these double strand breaks will be repaired by our machinery, because whenever our machinery finds that there is a break in the DNA, it will see that how to repair it. So if sister chromatid or homologous chromosome is available, then it will do homology based repair. And if no such chromatid or no such similarity is found, then it will go for non-homologous end joining. So in case of non-homologous end joining, as we have seen, it can lead to gene disruption. So after the break has happened, so there could be loss of some of the nucleotides which are present near to these breakage site. So due to which it will lead to gene disruption. That means if some gene is present and I am cutting from here, then the region of the genes, because some of the nucleotides get deleted. So in that case, gene will get disrupted. Gene will not properly give rise to the proteins. Similarly, in the case of homology directed repair, we can provide the sister chromatid information, or we can provide the template strand with respect to which it can synthesize the new strand. It can join these broken strands together. So we can add the donor DNA. We can add gene of interest into it, so which can lead to gene correction or gene addition. So this is how we can do the genome modification with the help of homology directed repair. Yeah, and similarly here also it is shown that uh, we can also induce these breaks by using the TELN. So this shows the zinc finger nucleases. Similarly, these subdomains can also be TELN based subdomains, and this can also lead to both non-homologous end joining and homology directed repair. And in case of non-homologous joining, it leads to gene disruption. In case of homology, it can lead to gene correction and gene addition. Okay, now we'll talk about the zinc finger domains, because we have looked into the zinc finger domains that these are present in the case of FOC1 nucleases, three subdomains which we have seen. So in 1940, the role of zinc 2 in enzymes was studied. So specifically carbonic anhydrase, when it was being studied, then they realized that this zinc ions, they are very important for the activity of this enzyme. They have seen that the enzyme is cooperating with these ions. So in 1985, Xenopus levis organism was studied, in which transcription factor 3A was discovered, which was showing the repeated 30 amino acid zinc finger binding motifs. So these motifs were present in this particular protein. So in 1985, it was evident that how the zinc finger domains are present inside the proteins. So if we talk about the structure of these zinc finger domains, then each finger, so as you can see, these different colors represent different fingers. So in this case, we have three fingers, and each finger, if we talk about one single finger, so this green region, it shows one single finger. So this particular is composed of 30 amino acids, which fold around the zinc 2 positive ion, which is coordinated by cysteine and histidine. So when we were talking about different families, then we looked into the C2H2 zinc finger motifs. So similarly, these zinc ions, the interaction is between the cysteine and histidine with the zinc ion. Yeah, so this represents the alpha helix and two beta sheets. And this one single unit is around 30 amino acids in length, and this is the zinc finger motif. And in these alpha helices, there are some of the region which is the two cysteines, and the next slide maybe we'll see that where the cysteine and histidine are present and how it interacts. So in this case, it interacts with the zinc ion in between. Yeah, so in this case, these are the cysteine side chains. Uh, yeah, so these are the cysteine side chains, two cysteines and two histidines. They're interacting with the zinc ion. Okay, now typical motif is beta hairpin, which is the two beta strands are present. So because these two beta strands connected together with the turn and they're anti-parallel to each other, so we'll not call them as a beta sheet. This particular is also a smaller motif, which is our beta hairpin motif. So keep this thing in mind that motif is the smallest structure which keeps on repeating. So smallest but not alpha helix and beta sheet, means it has multiple occurrences of those secondary structures, but after combination they are the smallest ones. So for example, in this case we have two anti-parallel beta sheets which are joined together with the help of a smaller turn. So this particular structure in itself is a beta hairpin motif, and then this beta hairpin motif is connected to the alpha helix, and this they together form the structure which is the beta beta alpha fold. This is known as uh, finger of the zinc fingers, one of the finger among zinc finger motif. And if we talk about conserved residues, then cysteine, histidine, which are conserved for the zinc binding. It has a hydrophobic uh, core residues which stabilize this fold. So core means which are present in the center of the protein, and hydrophobic means which repel water. So there are some amino acids which are hydrophobic in nature. So those amino acids are present in the core region of this particular zinc finger. Yeah, and one more thing which I want to mention is that they have mentioned conserved residues. Conserved residues means which the residues which remain fixed. So this shows you that in different proteins, the sequence can change, but still they can form the zinc finger motif. Because it is not that alpha helix can only be formed if given amino acids are present. There are multiple amino acids which can give rise to alpha helical structure. So in this case also, different zinc finger motifs can vary in their amino acid composition, but the residues which remain conserved are the cysteine and histidine. So other amino acids can vary, but they vary in such a way that they do not change the structure. Alpha helix is maintained. Okay, and the hydrophobic core is also maintained so that it remains stabilized. Because if it is not hydrophobic, then it will make on interactions with other components of the protein and the structure will get disturbed or it become destabilized. And whenever we say zinc finger domains, then in this zinc finger domain, multiple zinc finger units are present. Generally, at least three zinc fingers are present in one zinc finger domain, and there could be multiple such zinc fingers that could be present. So this shows that how these zinc fingers are connected to one another, and these alpha helices which are present in each zinc finger, they are responsible for binding to the DNA. So zinc finger domains, they are responsible for the binding to the DNA. And now we are not talking about the protein. Zinc finger domain is like a structural component. So domain is a three-dimensional structure of a protein which in itself is functional. So domain is a part of a protein. Protein is a big entity in which multiple domains can be present. So here we are only talking about the domain region of a bigger protein. So in big protein, these domains are also further broken into two beta strands and one alpha helix. And alpha helix component of this particular motif is responsible for binding to the DNA. So we can say that zinc finger domains are responsible for binding of protein to the DNA. Okay, now we'll see the diversity. So these zinc finger domains are also diverse. They represent 3% of the human proteome. So if we look into the entire proteins which are present, around 3% of them are the zinc finger domains, include the zinc finger domain structure. And they are most common eukaryotic DNA binding motifs. And the function is DNA recognition, transcription regulation, and protein-protein interactions. So now, according to the Krishna et al. Study of 2003, they have classified these zinc finger domains into these classes. So one is C2H2 like finger, which we have studied that how two cysteine residues and two histidine residues. Okay, so now here you can see. So one cysteine residue is present in this turn and another is the initiation of another beta sheet, whereas two histidines are present in the alpha helical structure. So they coordinate with the zinc 2 ion, due to which the name is C2H2 like finger. Another could be gag knuckle. So this is how the structure will appear. This is the gag knuckle. Then treble clef. So this is the how the structure of treble clef look like. This represents the limb domain. Then Zn2 cysteine 6 like finger. Then Tas2 like short zinc binding loops and metallothioneins. Okay, so now because they all are responsible to coordinate with the zinc ion, but their structure can vary. So based on that, these are the different classes. So it's not like they will always appear in the form which we have seen previously. They can appear in different forms. So all of them vary in their structure, but still they all come under zinc finger domains because they are responsible for the coordination with the zinc ions. Okay, now we'll mainly focus on the C2H2 type because these are the most common zinc finger nucleases or zinc finger domains which are observed in the humans. So each finger recognizes the three base pairs in the major groove. Now keep this thing also in mind because it keeps on repeating. So majorly in DNA strand, the region which is mainly identified as the major groove. And specific recognition depends on the amino acids at the alpha helix positions, which is -1, 2, 3, and 6. So when we were talking about this particular structure, so what -1 means that, for example, if my alpha helix is starting from this residue, so this will be my first residue. So the residue which is before this is my -1 residue. So -1 means upstream and positive means downstream. So whenever if these terms are used, then don't get confused. So whenever we are talking something and we are talking before it, then it is upstream. So in this case, -1 means the residue which is present one position before the start of this alpha helix. So the residues which are responsible for identifying the DNA or binding to the DNA are the -1, 2, 3, and 6. So they recognize the DNA strand. And binding modes, so non-specific binding, which means initially the electrostatic attraction to the DNA backbone. And then specific binding, so sliding along the DNA, fingers insert alpha helix into the major groove at the target site. So this is how binding takes place, and this binding is electrostatic interactions. So this is non-specific binding initially, and then after sliding through the DNA, it becomes specific. So which means that as soon as this particular zinc finger motif comes, it will not directly bind to the region. Firstly, it will non-specifically bind to DNA, and then it will move till the point where it will identify its recognition sequence where this particular alpha helix could bind to those three nucleotides. So the crystal structure was given by Paveltic and Pabo in 1991, and the protein name is ZIF268, in which there are three zinc fingers. So, as I mentioned, that zinc fingers are not proteins, these are the domains or motifs. So, in multiple proteins, these domains can be present. So, similarly, the crystal structures can also vary depending on which protein you are studying and how many zinc finger domains are present. So, this Paveltek, when he was studying the ZIF268 protein, so in that case, there were three zinc fingers which form a semicircle in the DNA major groove, and the amino acids at -1, 2, 3, and 6, which is critical for the base recognition. So similarly, which we have seen that these residues are responsible to decide that to which region in the DNA they'll bind. And if we talk about the engineering, that whenever you are designing them for specific purposes, so by altering these residues, zinc fingers can be designed for sequence-specific DNA binding. So as I know that in this alpha helix, these are the sites which are responsible for binding. So based on which DNA region I want to bind, I can change these sites. So that is what is mentioned here, that I can alter the residues at the protein level, which can uh, now specifically bind to the DNA region. So when fused to nuclease, which is the fork one, they become programmable genome editing tools. So these ring finger domains, they are only responsible for DNA binding. There is no other functionality linked to them. So if I want to bring some functionality, so I can link them to some nucleases such as fork 1. So with the help of fork 1, the C terminal region of fork 1, they can also bring about the cleavage. Okay, now discovery of DNA recognition code. So how it has been identified that where it binds in the DNA. So the same scientists, Paveltic and Pabo, they have solved the crystal structure for this particular protein, in which they have seen that there are three zinc finger proteins are present, and each finger contacts a three base pair DNA subsite. So why subsite? Because it is a smaller site. So three base pair DNA subsite is identified by them. So this is how it has been shown. So the 5' strand of DNA. So we know that nucleases work in the form of dimeric form. So in this case, I'm only representing it in the monomeric form, that how the DNA contacts are made. So from 5' to 3' strand, this is only the smaller region of DNA is shown here. So my finger 1, it will bind to GCG, whereas finger 2 is binding to GGT. Finger 3 is binding to GCG. Now, how it is binding? So these are the residues. These are the protein residues which I mentioned, REDR. So R means arginine. Arginine is one of the amino acid which is present in the protein. So we, as I mentioned, that -1, 2, 3, 6. So in the alpha helical region of the zinc finger domain, at these positions, these residues decide that to which DNA nucleotide it will bind to. So in this case, arginine, D stands for the aspartic acid. These are one letter code for amino acids. And these amino acids, so aspartic acid, these are generally negatively charged, whereas arginine is positively charged amino acid. And E is the glutamic acid. This is also negatively charged, whereas arginine is positively charged. So these are charged amino acids. So also remember this thing in mind, that because these are present in the alpha helical region and they are charged, so due to the charge on these amino acids, the interactions which they make are the electrostatic interactions. Okay, so in this case, this is threonine, which is T, H is histidine, D is aspartic acid, and R is again arginine. Okay, so these residues binds to TGG. So if we change these amino acids, we can change the region in the DNA also. So that is how the engineering can be done. We can decide to which part of the DNA we want the zinc finger domains to bind. But this is just to show you that in which sites we need to make changes. And these are the three nucleotides which are recognized by these amino acids. Okay, so it led to the realization that arrays of zinc fingers could be engineered to recognize the long DNA sequences, enabled the development of polydactyl zinc finger proteins with the high specificity. Okay, so in this case, there are only three fingers, but we can also engineer them. We can join multiple fingers. In that way, we can bind our protein to multiple or the long stretch of DNA. So after this study, it has been observed that we can design those as well. Okay. So now how to develop these zinc finger arrays? Arrays means when we are repeating something into the longer units. So that is referred to as array here. So modular assembly strategies emerged, which means we can build the arrays from the pre-characterized fingers. But the problem is lack of standardized reagents and protocols, and adjacent zinc finger modules influence each other. So it is context, there is a context effect. So Wright et al., these are the group of scientists in 2006, they have created archive of 140 plus zinc finger modules plus web tool which is Zipit to find the zinc finger target sites. So from here we can see that which zinc finger will bind to what target site. So after knowing this, we can design, we can combine multiple zinc fingers, and we will be able to decide that to which DNA it can bind. So now these group of scientists, they have ease down the process. Previously, there was no standardization. You didn't know that how the zinc fingers will affect, how combining different zinc fingers can influence to which part they will bind. So now it is well studied, it is present in the database. So now we can look into that. Bacterial reporter assays allow the screening of zinc finger binding in 24 to 16 days. So we can also test them, and we can check in the bacteria that how it is working, whether they are able to precisely bind to that region or not. Add gene hosts, resources, and plasmids for the zinc finger construction. So this is another server which also shows you different resources and plasmids where we can construct the zinc finger, because in the plasmids, if we construct them, we can check that what is the how whether these zinc fingers are working precisely or not, because we have seen that they have context effect. They can influence the binding of other means joined zinc fingers. So all these things can be studied with the help of plasmids. So in case of modular design means where we are joining multiple zinc fingers to each other, and these zinc fingers, they have different binding specificities to the DNA. So we can decide that which zinc fingers we want to link to each other. And the group of amino acids which are responsible for this linking is referred to as a linker, which is joining my two different zinc fingers. Yeah, and this is how it is shown that this is the three zinc finger protein which recognizes nine base pairs. Now, in this case, we have joined two different units of three finger ZFP. So in this case, we have now six zinc fingers. Similarly, we can also join the group of two zinc fingers. So there are three separate units of two zinc fingers. So there are total of the six zinc fingers which are present here, which recognizes the 18 base pairs. So depending on our need, we can design these arrays of zinc fingers. So this is modular assembly. I'm joining them one after other. Now creation of artificial nucleases. So in 1996, Kim Cha and Chandra Chandrasekharan, he created first artificial site-specific endonucleases by fusing zinc finger proteins with the FOC1 cleavage domain. So how we can uh, increase the functionality of nucleases. So nucleases have simple work, they can cleave the DNA. But yeah, and they also have the recognition site. But how they will bind to the DNA? So for that region, we need some of the domains or some motifs in the protein which can recognize the DNA. So in this case, to design the artificial nuclease or to design the site-specific endonuclease, they have fused zinc finger proteins with the FOC1 cleavage domain. So when we look into the FOC1 domain, it has two regions. So one region, which is the N-terminal domain, which is responsible for binding to particular region. So if we remove that N-terminal region and replace it with our choice of ZFPs, then we can decide that where this fork one will bind, where it will recognize. So in this way, this is our artificial site-specific endonuclease, where we are artificially selecting the site. And these engineered nucleases cut the DNA at predetermined sites, which we have decided. So this led to the foundation of zinc finger nuclease technology, in which we can decide where to cut, and later on we can make different modifications. We can insert our gene of interest, we can disrupt the gene which is already present, or multiple things can be done. We can correct the gene which was previously defected. Yeah, and this shows the same. So this shows the process that how we can design the artificial nucleases. So these circles represent different zinc fingers, and then there's a phage library. So all of these zinc fingers, they are tested, they are incubated, and then they are washed away. So it is observed that whether they remain together or not. So there are some cases where we are linking different zinc fingers by the help of linkers. So after washing, we'll see that whether they remain together or whether they get washed away. So in this case, as you can see, there are only two zinc fingers and one has got washed away. So these studies are done, and after that, after washing, the unbound or the properly available zinc finger domains are taken. They are incubated with the target DNA, because these zinc finger are responsible for binding to the target DNA. So we will isolate the high affinity zinc finger for the target DNA. So out of all these bacteriophages, the one which binds more tightly to our target DNA will be one which will be considered. So now we will recovery and amplification of the selected zinc fingers, and now we will clone the library of these zinc fingers. So what I am doing here is I am finding that which three main zinc fingers will bind better to my target DNA. So in this case, I am finding those zinc finger domains and I'm artificially constructing it. So this is the test where I will find out that from all the zinc finger domains which I have constructed, or the zinc finger arrays which we have constructed, which one is better, which one can bind better to the target DNA or to the target site. So this is where we are testing it, and in the end we have obtained that which one is better, so we can make a library out of it, and we can also study for the other zinc finger proteins that to which region they bind. And if we are interested in that, then we can add the fork one nuclease to it, and we can use it for our DNA or the genome on which we want to see its function. Okay, now so this shows you the zinc finger nuclease. Now this is the cleavage by homodimer. That means in both of them we have same units, we have same sites which recognize and binds, whereas it can vary also. So as in this case, this 1, 2, 3, these are the three subdomains which are present. So most probably here it is the zinc finger domain. Now these 1, 2, 3, these zinc finger domains bind to different uh, nucleotides. So we can design these heterodimeric kind of zinc finger nucleases as well. This is about the homodimeric and heterodimeric, depending on that where the sites it should bind. Okay, now this is the engineering platform. So modular assembly we have already seen, in which we have seen that how we can join different uh, zinc fingers to each other with the help of linkers. Similarly, there is another assembly which is the context dependent assembly. So context dependent means that you know already the context, what is the purpose, why you are designing it. So based on that, if we assemble these zinc finger nucleases, as we have seen in this case, that we know which are the sites against which we want to design. This is like context specific. Okay, so we know the interactions where we want these zinc fingers to interact. So this is how the context sensitive selection is done. These are different zinc fingers which are joined to each other in the form of three zinc fingers, and out of them we shuffle them and we select that which one can better bind to my DNA region, which is my site of interest. Okay, now it is publicly available platform with higher success. Sender et al. In 2011, he has successfully used this context-dependent assembly, zinc finger nucleases in zebrafish, Arabidopsis, and soybean, and he has produced the zinc fingers with high activity and low toxicity. Now, the third type of method where the zinc finger nucleases can be designed is oligomerized pool engineering, which is also known as open, in which combinatorial selection based and open source method is used. It uses bacterial 2 hybrid system for screening, and it relies on pre-characterized zinc finger pools assembled by PCR and then screened for the strongest binders. So all these approaches are nearly similar and higher success than the modular assembly, but it is labor intensive. So in modular assembly, we are simply joining them. We are not looking that whether it is binding or not. It is simply joining them together. In context dependent, we are also looking that uh, to which site we want to bind, and then we are joining it. Whereas in case of oligomerized pool, we were seeing that which one is specifically binding to the, which is strongly binding to the target site. So this is the open, and it has higher success than the modular assembly, but because we are looking into so many parameters, so it is time consuming. Okay, so now after the zinc finger nucleases have been designed, so next step is the DNA repair, because the nucleases is making their double-stranded cut. So now in after repair, there could be three outcomes which are possible. So if there is no homologous end joining, then it will lead to gene disruption, and disruption where we have done the cut. So at that site, the gene, whichever gene which was present, it gets disrupted. But if it undergoes the homology based repair, then one chance is that we can correct the gene. So we can supply the donor DNA and we can insert that, because for these arms, so in the previous session, if you remember, so how homologous recombination takes place. So there is the concept of homology arms, and whatever is present in between those homology arms, it get shuffled, it get recombined. So in this case, we can design some extra element in which we will keep our donor DNA in between the homology arms, and so that homologous based repair can work here. So in that case, we will replace, we will add some new gene, so we can correct the gene if there is something missing in between the genes. So with the help of double strand breaks, we can degrade it and we can add the new gene which is now functional, or we can also add the new gene. So these are two separate outcomes. Okay, so non-homologous end joining, as we know, it is error prone because it can introduce insertions, deletions, and it is generally used for the gene knockouts where we disturb the functionality of a gene. Whereas in case of homology directed repair, we either do gene correction or insertion. So double strand break induction is the rate limiting step for the HR based gene editing. Okay, this is the review. So if you're interested, you can go through this review in which they have explained that how after the double strand break, what are the fates that through which the DNA can pass. Okay, now zinc finger nuclease mediated gene targeting. So this is also showing the similar thing, but we are adding some more details. So in this case, what we does is we deliver the zinc finger nucleases to the cells. Now we are seeing that how in the cells we are doing it. So zinc finger nuclease. Now keep this thing in mind that zinc finger domain and zinc finger nuclease. So zinc finger domain is a smaller structure of a big protein. But when we say zinc finger nuclease, we mean that zinc finger domains are present, but nuclease region is also present. So this will be my protein such as fork1 nuclease. Similarly, zinc finger nuclease means in which I have a cleavage domain, which is the C-terminal domain of fork1 nuclease. And in case of N-terminal region, I have zinc finger subdomains. So that together is referred to as zinc finger nuclease. So we can deliver zinc finger nuclease into the cells. Now in the cells, we have the chromosome, we have the DNA. Now we have designed the zinc finger nuclease in such a way that they bind to specific region in the DNA. So it will introduce the targeted double strand break in our gene of interest. So for this, we must know what is the gene of interest where we want to make the double strand break. Now repair by NHEJ and HR. So NHEJ is the mutagenic knockouts. Now how we can make sure that homologous recombination based repair takes place. So in that case, we can supply the donor DNA. We can supply the uh, our trans means our DNA, in which it will be having the homology arm and the donor DNA in between, so that the strand which has undergone the double strand break, it can get recombined, homologous recombination takes place, and donor DNA get inserted into that particular strand. Okay, now it also depends that how you interfere with it. So you can add a donor DNA and you can also add some mutation into it. So in that case, you can bring about point mutation, or you can simply add some donor DNA. So that is referred to as gene replacement, in which you are replacing that particular region which was present here. Okay, so this leads to substitution, and whereas in case of NHEJ, it will lead to disruption, which we have already discussed so far. So for validation, you ensure that donor DNA not integrated elsewhere. So to ensure it, we perform the Southern blotting, and this is how the Southern blotting results appear. So you use a gel in which you enter, means one place is for the control and another well, or you can say, because this Southern blotting, it takes place in a gel. So in this case, we have different wells, we have different holes in which we put our sample, and then that DNA sample passes through the gel, it moves. So I'll not go into the complete details, but means if I talk in overview, then the bands which appears, it tells you what is the length of the fragment of DNA. So based on the size, particular DNA can move fast or slow in that particular gel, and then we take the picture of the gel to see that what are the different fragments which are present. So in this case, these are the corrected cells 1 kb and 0.7 kb. So for example, our normal DNA was 1.7 kb. So after the zinc finger nucleases, I have broken it into we have done double strand breaks. So now what we want is that it should break down into 1 kb and 0.7 kb. So whenever I will run it in the gel, then I must see in the control there will be only one band, which will be for the 1.7 kb size. But in my another strand where in my another region in the gel where I'm adding the sample after the zinc finger nuclease activity, I must see two bands. So these two bands will show that there are two fragments which are present. So this thing can be validated with the help of Southern blotting, that whether my zinc finger nuclease has properly cleaved it or not. And then with respect to the control, which is the previous on wild type sample or the normal sample, it should show some variable bands. If it is showing the same band, that means it is not working properly. And because in this case we are doing the site specific double strand breaks and we are deciding where to add the donor DNA, so based on that we already have the information that what will be my band size, or what will be the length of the DNA fragment appeared after the DNA repair. So based on that, we can judge it from the Southern blotting uh, maps. Okay, now this is the computational tool for the zinc finger nucleus design, which is ZIFIT, so which we have seen previously also. That it gives you the information of all the zinc finger nucleases and the sites where they can bind. So we can search the query DNA for possible ZFP target sites. So in this case, this shows that they have given a sequence of DNA. So I can give a sequence of DNA and I can submit it. So it will, in the result, it will tell me that where are the sites which are present where the zinc fingers can bind. So it will give me those sites, and then I can see that, for example, in this DNA, there is some gene where I want the zinc fingers to bind. So I can see which sites are available for the binding. So in that way, we can design particular zinc fingers to target that specific site. So it suggests available zinc finger modules from the archives, and it facilitates custom zinc finger nucleus design by linking modular databases with user-defined targets. Because we are defining the target, we are defining the region where we want the zinc fingers to bind. So if you're interested, you can go through this article, which is about the ZIFIT, and this is how it appears. So right now, when I was looking into this ZIFIT server, then it is not working. But yeah, this screenshot I've obtained from the already published paper where they have shown this. So there are some tools which might not work today, but this is the basic idea why they were designed and what the information they can provide you. Okay, now till now we have seen that how zinc finger nucleases work and how we can design them, how we can manipulate the DNA, and how we can identify that which zinc fingers we should combine to each other. So now then comes delivering strategies, because we want them to function inside the cell. That cell could be mouse or human. So how we can deliver them into the cell? So there are different strategies with which we can deliver them. So first one is the viral vectors. Another is non-viral vectors. C is the physical methods, and D one is the direct protein and delivery. So viral vector means we can use the adenoviral vectors. So these are the viruses which has larger capacity, but they are immunogenic. That means as soon as we put them into the individuals, then they will generate the immunogenic response in which our body will synthesize antibodies against them. So it is used in the HIV ZFN therapy trials. Then the lentivirals, so they can penetrate into the nuclear membranes and they can accommodate around 10 kb of whatever uh, means zinc finger nucleases we want to attach. But there is risk of insertional mutagenesis, and IDLVs are safer. Okay, so now these are some of the limitations also which are associated with them. And then comes AAVs. So these are low immunogenicity. They have high efficiency, but the protein which they can integrate is of the small capacity, which is around 4.7 kb, which can carry ZFN genes plus donor template. Okay, now what I want to mention here is that in this case, we are looking into the delivery of zinc finger nucleases. So viral vectors, what they will do is viral vectors, because we have seen plasmid vectors, phages, and all those things. So viral vectors is like in which viruses are there. So these are more likely the phage phage vector, but they are not infecting the bacteria. They can be any other virus. So that's why they are referred to as viral vectors. And we are integrating the zinc finger nucleases. So now we cannot integrate the protein into DNA. So what we does is we use the genes of zinc finger nucleases, which these genes, as soon as they enter into the host cell, then they will get um, they will means first of all they will replicate, they will make their copies, they will transcribe into the RNAs, and then they will make proteins, which will be my zinc finger nuclease protein. So with the help of viral vectors, I can insert the gene which contains zinc finger nucleases along with the donor template. So this donor template will come into play when the double strand breaks are done, and after that the homology repair will take place. So in that case, donor template will make sure that your gene get inserted into the place where the double strand break has taken place. So I can add new gene, I can correct the already available gene. So all those things will be done by the donor template, and zinc finger genes will be responsible for the expression of zinc finger nucleases protein, which will ultimately lead to the double strand breaks. Then if we talk about non-viral vectors, then there are polymers, liposomes, CPPs. So they can also integrate these zinc finger nucleases along with them, and they can deliver them into the cell. Then physical methods are microinjection or electroporation. So in case of microinjections, there is a small needle with which we can insert zinc finger nucleases directly into the nucleus of the cell. Then electroporation is based on the electric charges. We'll create the pores in the cell membrane, and then we insert these zinc finger nucleases into the cells. And if we talk about direct protein delivery, so in these methods, we are only inserting the genes of zinc finger nucleases, and then these genes will express, make RNA, then they will make protein. So direct protein delivery means we are directly delivering the protein. We are not delivering the gene. So this zinc finger nucleases, they can cross the membranes due to this C2H2 zinc finger domains. But the challenge is that these zinc finger nucleases proteins, they degrade within the 4 hours. So because our body has the nucleases and different proteases which degrade these proteins, so in 4 hours that protein will get degraded. So to overcome that degradation and to stabilize our zinc finger nucleases, the strategy is that we modify the lysine residues which are used to stabilize the protein, so which act as a stabilizer. Okay, so these are the delivery strategies which are involved in the delivering of the zinc finger nucleases into the cell. And this is the visual representation. So this is obtained from the this particular site. So if you're interested, you can go through this site. This also shows the same thing, but it shows you some additions. So there is cell squeezing technique which can also be used. Nanoparticles can be used. So in nanoparticles, we can also attach these zinc finger nucleases into it, and then we can insert it into the cell because they can easily pass through the cell membrane. And then these are some of the example of the CRISPR-Cas9 method, in which, so instead of zinc finger nucleases, we can use CRISPR-Cas9, or we can use talons. They recognize the DNA and then they make the cleavage. So this is not, as of now, linked with the ZFNs. Mainly ZFNs, we are only dealing with this thing. And then the transposons can be used to insert the gene of interest. So there are different things which have been mentioned here, and we are right now only focusing on these ZFNs, and these are the delivery technologies which we have focused here. Okay, then comes fusion protein design strategies. So fusion protein means we are joining two separate proteins with one another. So these are the artificial construct in which we are joining domains from the separate genes. So this is the example of 2A peptides, which is 18 to 22 amino acid long. So this is the name 2A. And peptide means how it vary from protein. So protein is a bigger stretch of amino acid. So if it is a smaller stretch of amino acid, it is generally referred to as peptide. When the length is generally 20 to 30 amino acids. So these are the smaller length proteins. So they are also referred to as peptides. So they cause the ribosomal skipping during the translation multiple proteins from one mRNA. So, for example, we have only a single RNA. So, in that case, to design multiple proteins from that RNA, we can insert the 2A peptides. These 2A peptides, what they will do, as soon as ribosome reaches this site, it will now get removed. So, it will synthesize the protein till here. Next time, it will start from here and synthesize the protein 2. So in that way, one mRNA can give rise to two separate proteins by integrating this 2A peptide, which is responsible for the skipping of ribosome. So examples are F2A, which is obtained from foot and mouth disease virus, P2A, T2A. So this is the sequence of them. So these constructs, whenever we put them into the RNA, then they will make sure that as soon as ribosome reach these sequences, it will get removed, and same mRNA can give rise to multiple proteins. That is the main application of this fusion protein. It is used to co-express paired ZFNs from one construct. Okay, now with this we can means what we can do is that if I want that two ZFNs to get expressed in the cell and I only want to insert only one construct, so what I can do, I can link two separate proteins of zinc finger nucleases. Now what, when we say protein 1 and protein 2, we do not mean that it is a protein. We mean that it will give rise to protein 1. This is the mRNA sequence, which will give rise to protein 1 and protein 2. So what I can do is that in zinc finger nucleases, we require it in the dimeric form. So we can join both of them and we will keep one 2A peptides. So name is 2A, which will break it down. Whenever it enters into the body, this single mRNA can give rise to two separate components. So two monomeric units of zinc finger nucleases can be synthesized and then they can bring about the double strand breaks. So this thing can be done, that instead of adding them separately, I can join them together and just keeping one single 2A domain in between, so that whenever it enters into the body, it generates the pair of zinc finger nucleases and not a single protein. Okay, now this particular figure also um, highlights that how the process has been done. So key design factors. Firstly, we decide the insertion site, domain interactions, domain orientations. So all those things we keep in mind when we design these fusion proteins, that where we want to insert the domain. In this case, it is a 2A peptide domain. Then post-translational conjugation, that after the means after it converts into the protein, how it will conjugate together. So in the case of zinc finger fork 1 nuclease, when we were talking, so it's uh, C terminal domain, how it links, how it interacts with each other. So two uh, it makes a dimeric protein, and then how those dimeric units are interacting to one another. So it comes under post-translational conjugation, that after two proteins which are getting synthesized, how they will uh, interact with one another, whether they will cross link, or all those things are also kept in mind. And then tandem fusion. So tandem fusion simply means that two proteins, in case of zinc finger, I'm considering them to be same, but if it is my heterodimeric zinc finger, then both these proteins will also be different. So in that case, the tandem fusion means we are joining them next to each other We are linking them with the help of linkers. So this fusion means when we are joining two proteins, this is referred to as tandem fusion. And it can be between two different proteins also. So protein 1, protein 2, and it can be between same protein also. So okay, so this tells you about the how the fusion proteins are designed and what is the purpose of designing the fusion proteins. Okay, now we'll see some of the examples. So zinc finger nucleases targeting the CCR5 gene. Okay, now uh, this construct includes the zinc finger nuclease left, zinc finger nuclease right. So right side of the zinc finger nuclease is referred to as ZFNR. So this is our left construct and this is the right construct. And we know that these zinc finger nucleases, they work only in the dimeric forms. One single unit is not sufficient. So this is referred to as ZFNR and this is ZFNL. And they are binding to the CCR5 gene, which is the DNA. So this sequence is the is corresponding to this particular gene, and this fork one is binding to it, and it is cleaving this gene. That means if it will make double strand break, then this gene will not be able to make the proper protein, so it will get disrupted. So the application is in the HIV therapy by editing this CCR5 receptor in the T cells. And for the delivery of these zinc finger nucleases, the viral vectors are mainly used, which are adenoviruses, baculovirus, or lentiviruses. Okay, now in this case, the detailed picture is shown here. So each gene has different exons, and each exon combined together by the removal of introns, and they give rise to protein. So in this case, specifically when we use zinc finger nuclease, there is a site which is present in exon 3. After ZFN mediated double strand break, there is a cut here. Now, due to which this protein is now short and non-homologous end joining takes place, due to which this gene get disrupted. So some of the sequence get deleted, due to which there is disruption of this gene, so protein cannot be synthesized, and this CCR5 is responsible for generating a receptor, so that receptor will not be formed, due to which HIV, which uses this CCR5 receptor where it binds, so as soon as the receptor is not formed, so HIV will not be able to bind, and ultimately we can protect ourselves against the HIV attack. Okay, now there are some supporting technologies in the genome editing. So not just nucleases, but we also need the delivery agents of genome editing reagents such as zinc finger nucleases, donor DNAs, and then isolation of rare genetic variants for propagation, in vitro biology, cell tissue culture, transfection, transformation, regeneration. So this is just to show that not just nucleases, but some other things are also required in the process of genome editing. And the potential of genome editing is that it enables you to study the function of gene, how the gene functions, its mechanisms. You can design novel phenotypes. You can customize different species, customize them for different species and genomes. Then double strand break repair pathways. So with the help of homology dependent repair, we can edit the gene, we can insert the novel gene or new gene. And by NHEJ, we can do knockout studies or we can do targeted mutagenesis. Okay, now this is what is shown here. So other than zinc finger nucleases, TELN and CRISPR-Cas9 are also responsible for binding to the DNA specific sites. So TELN and CRISPR-Cas9, maybe we'll discuss them in later sessions, but right now we'll be focusing on the zinc finger nucleases, and this is the double strand repair mechanisms. So depending on both of the repair mechanisms, the fate can be different. After a double strand break, it can enter into different fates. It can lead to deletions, insertions, or corrections. Okay, now there is another review which talks about the integration of CRISPR-Cas9 with artificial intelligence for improved cancer therapeutics. So if you're interested, you can go through this, that how CRISPR-Cas9 is getting involved in the cancer therapeutics because it is bringing about double strand breaks with which we can disrupt the genes. So in case of zinc finger nucleases, when we were looking at the CCR5, so we said that how this gene makes a receptor which binds to HIV. So if this gene get disrupted, receptor will not be formed and HIV will not be able to bind. So similarly, with the help of CRISPR-Cas9 also, we can target those genes which are responsible for the cancer. So we can disrupt them or we can correct them and we can stop the cancer spread. Okay, now to quickly go through them, for example, if cancer, so how cancer happens, there are different regions for the cancer. So few of the reasons come under genetic and epigenetic aberrations. If there is uh, improper rearrangements, or if there is single nucleotide polymorphism, or if there is mutation or non-coding RNAs, histone modifications, alternative splicing, all these regions can be responsible for the cancer generation or cancer onset. So how we can cure it? So we can design the nucleases which can specifically target those sites where these things have happened. So that could be with the zinc finger nucleases or the talents, which are transcription activator like effector nucleases or CRISPR-Cas9. So in this paper, the focus is on the CRISPR-Cas9, but zinc finger nucleases can also work. So yeah, this shows you the genes which either get upregulated or leads to the mutations. So we can design strategies in such a way that we can target these genes, and after targeting them, we can change their functionality, we can modify their functionality and control the cancer. Okay, now yeah, these are the applications which have been mentioned, that how it has involved in cancer research. So it has brought about the epigenetic editing of cancer cells. It have led to detour virus-induced tumor regenesis, then drug development against cancer, targeted immunotherapy, diagnostics, generation of cancer models. So all these things are the applications of CRISPR-Cas9. Okay, now we'll quickly go through all the applications of zinc finger nucleases. So we can bring mutations by the gene deletions. Okay, now I'll not go into the detail, but there is a yellow gene in Drosophila which is responsible for the cuticle pigmentation. So its mutations can lead to the complete loss or the mosaic patterns in its abdominal part. Then there is a study by Bibikova et al. In 2002 in which he has designed these zinc finger nucleases. So this is the study in which he has designed zinc finger nucleases which target this particular Y gene, and it is expressed in the larval induced. It induces the somatic mutations. So in overview, what he has done is that these zinc finger nucleases, they bind these Y gene and they bring mutation. So to check that whether the mutation has happened, he checked the color of this cuticle. So if the color is like, if yellow patches appear, that means mutations have taken place. And if it is dark, which is the normal one, then that means mutation has not taken place. So according to this study, he has said that we can use zinc finger nucleases to bring about the mutations. So this is their main aim, and this start, why, and if you want to go into the detail, you can read this thing. I have mentioned everything. So in this case, mainly what they have done is they have changed the sequence of these zinc finger domains in such a way that they can bind to this yellow region, this yellow gene region. That is what is mentioned here. Yeah, so this study shows you that zinc finger nucleases have the potential to bring the mutation in the gene. Then second application is large gene deletions. So different scientists have performed study into this, Kai et al., Patulino et al., Voitas. So in their studies, they have shown that these zinc finger nucleases can also delete the larger genes fragments. So according to Kai et al., he has used the reporter construct with GFP fragments plus 2.8 kb sequence in tobacco. So he was studying the tobacco and he mentioned that ZFN expression can cause the targeted double strand break, recombination, and deletion. Similarly, Petolino et al. In 2010, he has also shown that 4.3 kb GUS sequence can be deleted with the help of zinc finger nucleases. So these are the sites where the break has happened with the help of zinc finger nucleases after the double strand break, and joining them back together with the nonsense mediate, with the help of non-homologous end joining repair method, we can cleave the larger fragment which was present in between, or we can also go through the homology directed repair, which can join this GFP construct together without the loss of the GFP signal. Yeah, so this shows you that zinc finger nucleases can also bring about the larger gene deletions. Okay, now this is the study by Petolino. So this is the paper in which he has clearly mentioned that how, with the help of zinc finger nucleases, deletion was achieved, and Means after the cleavage, if that particular double strand broke DNA, it can go into different methods of DNA repair. It can either go to the NHEJ, or it can go to the homology recombination based repair. So on the basis of that, we can bring about different modifications in the gene. Okay, so I'll not go into the detail. Then third application is the crop improvement. So one example of crop improvement is the study by Bonawitz et al. In 2019, in which he has used these zinc finger nucleases for the targeted integration at soybean FAD21 alpha locus. So this particular gene, in which he has made some insertion, and he has integrated multi-gene donors which were up to 16.2 kb. So that means with the help of ZFN, not just deletions but insertions of larger size can also be made up to 16.2 kbs, and this demonstrated a stable and fertile transgenic soybean plant. Okay, and these are the donor junctions which have been inserted into this FAD21A gene. Okay, so with the help of these insertions, the yield of this particular soybean plant has increased. It has become more fertile, more stable. So these are the advantages that we can also bring about the insertions with the help of ZFNs. Then gene replacements. So Bibikova et al. In 2003, he replaced the wild type Y gene, which we have seen in the previous example, that how he has bring about mutations in the yellow gene. So with respect to mutations, replacement can also be done. We can replace it with entirely new gene, and then we will see that how it behaves, whether the mutation is effectively showing up in the phenotype or not. So since then, it was demonstrated in multiple organisms, mice, rats, humans, and plants. So application is that we can create the disease models. We can see that how the mutations can lead to particular disease. So this is just for the yellow gene, but there are some genes which are responsible for the uh, patterns or some body structures in the organisms. For example, in Drosophila, wing structure or its uh, other parts. So with the mutation in those genes, we can see that how it influenced the organism, whether it leads to some disease or not. So with the help of zinc finger nucleases, we can replace those genes with the mutated genes, and we can see that how it appears, how it affects the function. So that's why it can create the disease models. Yeah, and similar thing is also shown here as well, that how different repair mechanisms can lead to different outputs. And in the previous session, we have looked into the negative and positive selection markers, that with the help of these markers, we can check that whether the recombination has taken place properly or not, whether it is the random recombination or it is site-specific recombination. So for those uh, reasons, we add the markers so that we can distinguish. Now, next application is in the HIV treatment. So when we were talking about CCR5 receptor, so it is a HIV co-receptor on CD4 positive T cells. So if we make the mutations in this particular receptor, then we will influence the protein which is responsible for the receptor synthesis, and we can stop HIV to enter our T cells. So one construct is CCR5 delta 32 mutation, in which there is a 32 base pair deletion. So in the previous example, when we were talking that how ZFN has led to the cleavage at the exon 3, so after that cleavage, if it undergoes NHEJ, then it is possible that some of the base pairs get deleted. So in this case, if there is 32 base pair deletion, and if these are homozygotes, then it is resistant to HIV-1. If it is heterozygotes, then it leads to delayed disease progression. These are the outcomes, that how this deletion mutation means consequence of this deletion. And clinical findings have suggested in 2009 the bone marrow transplant from the CCR5 del 32 donor cured an HIV patient, which was the Berlin patient. And limitation is that rarity of mutations and risk of BMT. And then ZFN strategy is means how zinc finger nucleases work. So they disrupt this gene in the patient's T cells. And then Manotham et al. In 2015, he has generated CCR5 stop codon in the MSCs using ZFN mediated HDR expandable clones for therapy. So different approaches can be achieved, not just by deleting, but by changing, by mutating it, or by inserting something in between. We can disturb this CCR5 gene expression. So based on that, different scientists have worked into it, and they have identified that if those variations are brought about in this gene, then it can control the HIV. And they have also seen that if we insert this deleted gene construct in the patients, then it can also cure them. It can cure HIV. So similarly, currently many studies are going on that how we can cure the cancer patients. So one application is in the cervical cancer, which is HPV related. So if we talk about cervical cancer, then according to 2020, around 6 lakh 4,000 new cases were reported and 3 lakh 42,000 deaths were there due to this cervical cancers. And this is mainly caused by the human papilloma virus, which is HPV type 16 and 18, which is around 50% of pre-cancers. So this HPV genome, there are three different regions in this genome. So one is early region, which is E1 to E7, which is responsible for the replication of this virus. Then there is late region, which is L1, L2, which is responsible for the capsid proteins. Because these viruses, their genetic material is surrounded by the protein coat. So that protein is synthesized by this late region, which is L1, L2, and then long control region, which is LCR, which contains the regulatory elements. So now the oncogene is this C6 and C7. So this early regions E6 and E7, it inhibits the P53. So P53 and PRB, these are the proteins. These are the tumor suppressor genes present in the humans. So these regions, they inhibit these genes, due to which they disrupt the tumor suppression and leads to the uncontrolled proliferation of cells and ultimately leads to cancer. So how the therapeutics can be designed? So firstly, antisense mRNAs were designed which could bind to these genes and they can repress these genes, or they can not allow the viral genome to express. So that is antisense strategy. Then ribozymes. So these are the enzymes which are composed of the ribosomes which can degrade those RNA molecules. Then small interfering RNA or small heterogeneous RNA strategies. These were the previous strategies which were designed to inhibit these regions so that we can control the spread of the cancer. But recent are the zinc finger nucleases, talins, and CRISPR-Cas9. So what they does is that in the this HPV virus, they make the double strand breaks in such a way that the region which is responsible for this binding to the tumor suppressor genes that get inhibited, that get affected, due to which it no longer binds to these uh, proteins, these genes, and ultimately it cannot lead to the cancer. So there are different scientists who have worked into it, Mino et al. In 2019 and 2013. So they have designed zinc finger nucleases against the HPV E2, which block the replication. So there are different ways. If we can stop the replication, then the virus will not make multiple copies. So that in that way also we can stop the cancer, or we can interfere the capsid proteins in which the protein coat will not be synthesized. So the nucleic acids which are present in this virus, it get degraded. So that way also we can control the cancer, or we can interfere with this LCR region in which regulatory elements are present, which are responsible for these component expression. So we can disturb these LCR regions so that the balance between them get disturbed. So in that case also we can stop the cervical cancer by influencing the genome of HPV. Okay, so these studies have shown that how different regions of this particular gene, if interfered, then they can lead to the cancer cure. They can inhibit the cancer cell growth and leads to the apoptosis. Okay, so these are some of the applications of zinc finger nucleases. Now we'll proceed to the MCQs. Okay, so the first question is, which of the following statement accurately describes the alpha helix secondary structure in proteins? Option A, the alpha helix is stabilized by the hydrogen bonds between the side chains of amino acids. Option B, each turn of alpha helix contains 5.4 amino acid residues and has a vertical distance of 1.5 angstrom between the equivalent atoms. Option C is The alpha helix has 3.6 amino acid residues per turn with a vertical distance of 5.4 angstrom between the structurally equivalent atoms in a turn. And option D is the alpha helix is the least common secondary structure and is stabilized by the ionic bonds. Okay, so yes, you are right because we have already seen that alpha helix has 3.6 amino acids per turn and the pitch is 5.4 angstrom. Yeah, and this is the structure which shows it. Okay, and then we have also seen that the hydrogen bond is formed between the NH group of one amino acid and C double bond O group of the another residue, which is one I plus 4 residue apart. Now, next question is which of the following best describes the function of the fork1 restriction endonuclease? Option A, fork1 recognizes symmetric nucleotide sequences and introduces blunt end cuts directly at the recognition site. Option B is fork1 belongs to a class of restriction endonucleases that recognize specific but asymmetric nucleotide sequences and introduce staggered cleavages at positions away from the recognition sequence. Option C is fork1 cleaves DNA randomly without recognizing specific sequences and create blunt ends. And option D is fork1 is a restriction endonuclease that recognize palindromic sequences and make cuts at the recognition site, resulting in blunt ends. Yeah, you're right. Because we have seen that it does not bring blunt end cuts. And in all the other three options, it mentions blunt ends. So with that reason, only every other option becomes wrong. And then we have also seen that it recognizes this site, but it cleaves some base pairs downstream. So 9 base pair downstreams for 5' site and 13 base pair downstream for 3' site. And this particular recognition site is also asymmetric site and it is non-palindromic. It is not palindromic in nature. Now, next question is which of the following features are characteristics of C2H2 zinc finger motif? Option A, it contains a repeated 20 to 25 amino acid sequence with a single conserved histidine and no cysteines. Option B, it is primarily found in RNA binding proteins and does not interact with DNA. Option C, it includes a 25 to 30 amino acid sequence with two conserved cysteines and two conserved histidines and is involved in transcription regulation and protein-protein interactions. Option D, it consists of a single zinc finger motif and functions solely in lipid binding. Yeah, so this is also straightforward because as the name suggests, C2H2, so it has conserved two cysteine and two histidine residues and it is 25 to 30 amino acid long. And this is the structural representation of it and how it coordinates with the zinc ion. Okay, next question is what is a key characteristic of the modular assembly method for zinc finger protein design? Option A, it uses a single zinc finger to target a 9 or 12 base pair sequence. Option B is individual zinc fingers are optimized against a target triplet DNA sequences and linked together to form 3 or 4 zinc finger proteins targeting 9 or 12 base pair sequences. Option C, it involves a context-dependent assembly of zinc finger arrays without optimizing individual zinc fingers. Option D, it employs a high throughput approach to design zinc finger proteins targeting long DNA sequences. So what is the key characteristic? Okay, so you mentioned option B, and yeah, it's right. So individual zinc fingers are optimized against the target triplet DNA sequences that we have seen, and they are linked together 3 or 4 zinc finger proteins, and then they target 9 to 12 base pairs because each zinc finger targets only 3 nucleotide or 3 base pairs. So that's why option B is right. And this is the representation of the modular assembly. Oh, sorry. So next question is which of the following steps is not involved in zinc finger nuclease mediated gene targeting? Option A, identifying zinc finger nuclease target sites near the targeted locus within the gene X of interest. Option B, designing or selecting zinc finger proteins that recognize the chosen zinc finger nucleotide target sites. Option C is converting the designed or selected zinc finger proteins into the zinc finger nucleases. And option D, applying a high throughput screening methods to test multiple gene editing outcomes simultaneously. Okay, now you have to tell which step is not involved. Yeah, so you're right. So applying a high throughput screening method to test multiple gene editing outcomes simultaneously is not involved in the gene targeting. Rest others are involved. Okay, so next question is which of the following is a key factor for successful direct delivery of zinc finger proteins into the cells? So this is the direct delivery. Option A, the low lipophilicity and the high pH sensitivity of zinc finger nucleotide proteins, which require modification to enhance the cell membrane penetration. Option B, the positive charge of cysteine to histidine to zinc finger domains in ZFN proteins, which aids in penetrating the cell membrane. Option C, high degradation rate of ZFN proteins, which necessitates frequent redelivery to maintain effectiveness. And option D, the use of non-enzymatic methods for protein delivery, which bypass the need for the stability modifications. Okay, so you have to mention what is the key factor for the successful delivery. Okay, now let's see. So the right answer is option B. So due to the positive charge on 2 cysteine and 2 histidine zinc finger domains, due to this positive charge helps them to penetrate into the cell membrane. Whereas if we look at the lipophilicity and pH sensitivity, so they are not the limiting factors for the ZFN delivery. And then while the ZFN proteins degrade quickly inside the cells, but that is a challenge and it is not the key factor in this delivery. And we have seen that how by modifying the lysine residues, we can make it more stable so that it does not get degraded. And then the non-enzymatic methods such as liposome and all those things, they apply to the nucleic acid delivery and not uh, direct delivery. But in the case, we've seen that, uh, but they do not bypass the stability modifications, because when we were talking about the non-enzymatic delivery by using the liposomes or all other things to deliver directly the protein zinc finger nucleases, then we have seen that the limitation is that as soon as it enters into the cell, it can undergo degradation. So there is a need for the stabilization by modifying the lysine residues. So that's why this option is also not right. Okay, next question is what is the primary function of ribosomal skipping in translation? So option A, it enhances the ribosome's ability to bind to the multiple mRNA molecules simultaneously. Option B, it allows for the incorporation of additional amino acids into the growing polypeptide chain. Option C, it prevents the ribosome from linking a new amino acid, leading to co-translational cleavage of the polyprotein. And option D is it ensures that the ribosomes only translate viral peptides and exclude the cellular proteins. Yeah, so we have seen that when that A2 peptide was inserted between the two proteins, so the main purpose is that we can, from a single construct, we can design multiple proteins. So we can undergo the co-translational cleavage from where multiple proteins can be synthesized from one single mRNA. Okay, next question is which repair process is utilized by the zinc finger nucleases when a donor template DNA is present? Yeah, so this is also straightforward. Option A is NHEJ, which is leading to the targeted mutagenesis. Option D is homology directed repair, leading to gene editing and site specific integration. Option C is error prone repair, resulting in the random insertion of nucleotides. And option D is translation synthesis, which bypasses the DNA damage without repair. Yeah, so this was straightforward because donor template was available, so it can undergo an HDR, which is homology directed repair. Now, next question is which of the following statements about the CCR5 chemokine receptor is true? Option A, CCR5 is a receptor with 5 transmembrane domains and is primarily expressed in liver and lungs. Option B, CCR5 functions solely as a receptor for signaling molecules in the immune system and does not interact with HIV. Option C, homozygosity of a 32 base pair deletion in CCR5 gene, which is del 32 or delta 32, confers strong resistance to HIV infection, while heterozygosity results in the slower HIV progression. Option D, CCR5 is expressed only on CD4 lymphocytes and does not have any role in the central nervous system. So this also we have seen. Yeah, so we have seen that how that particular deletion is responsible for the resistance, and it can also cure the patients who already have the HIV. Okay, so next question is which of the following statements about the cervical cancer and HPV is true because we have seen that HPV is responsible for the cervical cancer. So, option A, cervical cancer is most common in high income countries with fewer cases reported in low and middle income countries. Option B, HPV types 16 and 18 are responsible for less than 10% of high grade cervical precancers. Option C, human papilloma virus, which is HPV, is responsible for more than 95% of cervical cancer cases. And option D is Shankar and Associates used a four fingered compose ZR zinc finger nuclease pair to target the E6 gene of HPV16. So you have to tell which one is the true. Yeah, and this question is bit based on the facts. If you have read them, then only you can answer it. Okay, so this is a bit complicated one. So the right answer is option C, in which the HPV causes greater than 95% of cervical cancer cases, which makes it the main causative factor. Whereas if we talk about the first option, then the cervical cancer is actually more common in the low and middle income countries as compared to the high income countries. So the statement was not right. Similarly, in the second option, they have mentioned that HPV type 16 and 18 are responsible for less than 10% of high grade, but actually it accounts for 50% high grade cervical cancers and not less than 10%. So this option was also wrong. Whereas if, in this case, Shankar and Associates, so they have used six fingered ZFNs and not four fingered. So that's why this option is also not right. Okay, now I have 10 more questions. So if you have time, then I'll go through these 10 questions, or otherwise I'll keep them in presentation and you can practice them whenever you go through the presentation. So if you have time, then I can go through these questions. Otherwise, I'll stop here. Okay, so you can, whatever you want, you can type that in your chat box so that I can see that whether we should go through these 10 questions or okay. So thank you. We can go through these questions. So question 11 is, which of the following best explains why FOC1 must dimerize to cleave the DNA in zinc finger nucleases? Option A, because each monomer independently cuts one strand of the DNA. Option B, because FOC1 cleavage domain is catalytically inactive as a monomer and require dimerization for activity. Option C, because dimerization ensures zinc finger proteins can bind palindromic sequences only. And option D, because FOC1 dimerization prevents the random cleavage of genomic DNA. Okay, so this also we have discussed. Yeah, you're right, because we have seen that it is inactive if it is in the monomeric form. So that's why dimerization is important. So next question is, which is a key limitation of modular assembly in ZFP design? Option A, it cannot target more than 3 base pairs per zinc finger. Option B, context effects cause reduced binding efficiency when multiple zinc fingers are linked. Option C, it requires viral delivery method to function. And option D, it exclusively targets the RNA rather than DNA. Okay, so the right answer is option B. Because in modular assembly, we have joined multiple zinc fingers. So we can target more than 3 base pairs uh, per zinc finger. Okay, so this option is bit not written properly. So it cannot target more than 3 base pair per zinc finger. So this is true that one zinc finger can target 3 base pairs, but in the case of modular assembly, because we are joining multiple zinc fingers, so it is not just limited to 3 base pairs. Yeah, so this is means this point is true, but this is not the key limitation. The key limitation is that because if we are linking multiple zinc fingers, then how their linking can influence the binding based on the context. So that idea is that efficiency, binding efficiency get reduced. So which is the context effects. So this, when we were talking about it, then we have discussed it. And whereas the next, which it requires viral delivery, so this is not true. We can use the other delivery methods also, and it mainly target DNA, not RNA. So that's why this option is also not true. So next question is, which delivery method offers the largest transgene capacity but is highly immunogenic in human applications? So option A is adeno-associated virus, option B lentivirus, viral vectors, adenoviral vectors, and option D is direct protein delivery. So largest transgene, but which is also immunogenic. So adenoviral vectors is the right option because we have seen that it has largest capacity, but because it will lead to the immunogenic response, so we does not prefer it much. Okay, so up to 36 kb insert can be done, and direct protein delivery cannot be right, so it is completely wrong. Whereas the lentiviral vectors, so they are means inserts can be up to 8 kbs, whereas for the case of adeno-associated virus, the insert size is 4.5 kb. So adenoviral vectors is the right answer. Next question is, which of the following is true about the context-dependent assembly for the zinc finger nucleases? So option A is it does not consider interactions between the neighboring zinc fingers. Option B, it is less efficient than modular assembly. Option C, it accounts for context effects, increasing the success in designing the functional zinc finger nucleases. And option D is it is used only in the mammalian cells and not plants. Yeah, so this is also straightforward. Answer is the option C because as the name suggests, context dependent assembly, so it accounts for the context effects. So that side is better than modular assembly. Next question is what is the role of ZIFIT in ZFN research? So option A, it delivers ZFN proteins directly into the plant cells. Option B, it screens for the homology dependent repair versus NHEJ events in the edited cells. Option C, it identifies possible zinc finger binding sites in the target DNA sequences. And option D, it serves as a viral vector for ZFN delivery. Yeah, so this is also straightforward. We have already seen it. So next question is, which plant genome editing achievement demonstrated ZFN mediated integration of a multi-gene donor into the soybean FAD21A locus? So option A, Bibikova et al. A 2002 study, Bonawitz et al. 2019 study, Boetas 2013, and Petulino et al. 2010. So these kind of questions, I don't think they'll come in the exam because this is completely based on these studies. But just for your idea, I've kept this. Okay, so I'll tell. So the right answer is option B, which is Bonawitz et al. In 2019. So he looked into the soybean, and this shows you the application of ZFN in agriculture, that how we can enhance the stability and the yield of one particular uh, crop or one particular plant by making the integration of multi-gene donors into it. So this is the study. Next question is, in ZFN mediated genome editing, which repair outcome is typically exploited for the gene knockout? So homology directed repair, non-homologous end joining, base excision repair, and nucleotide excision repair. Yeah, so this is also straightforward, and we have already seen that how NHEJ leads to gene disruption. Next question, which of the following is not a typical challenge with direct protein delivery of ZFNs? Option A, rapid degradation of ZFN proteins after entering the cell. Option B, difficulty in penetrating the cell membrane. Option C, cytotoxicity caused by the lysine modifications used to stabilize ZFN proteins. And option D is short half-life of delivered proteins. So out of them, you have to tell which is not a typical challenge. Yeah. So this is not a challenge because lysine modifications are generally not toxic. So they are not cytotoxic. That's why this is not the typical limitation, whereas all other are the somehow the limitations of the protein delivery of ZFNs. Next question is, which clinical approach inspired ZFN-based disruption of CCR5 gene for HIV therapy? Option A, gene therapy using AAV, which is adeno-associated virus for hemophilia. Option B, the discovery that CCR5 del32 homozygotes are resistant to HIV infection. Option C, siRNA-based silencing of HIV structural proteins. And option D is development of HPV vaccines against cervical cancers. Okay, now let's see. So the right answer is option B because we have seen that after the 32 base pair deletion in the CCR5 gene, the if there are homozygote present, there is the resistance to HIV. So different studies have mentioned that because if it is resistant to HIV, so it became as an inspiration with which more and more people began working with this, and they have looked into that how with the help of ZFN we can interfere with this gene and we can lead to the HIV therapy. Okay, so last question is which of the following statements about the large gene deletions induced by ZFNs is true? Option A, ZFNs can only delete DNA fragments up to 2 kb. Option B, Patolino et al. 2010 reported successful deletion of 4.3 kb GUS sequence in plants. Option C, Voitas 2013 concluded ZFNs are not capable of large deletions. And option D, large deletions require both ZFNs and CRISPR-Cas9 in combination. Yeah, so all other options are completely wrong, and this option is right because we have seen. So I've also attached the paper. If you're interested, you can go through this paper in which they have mentioned that where the deletion of this GUSA gene has been done. So they have seen that this zinc finger nucleases, they can also lead to the larger deletions. So this is the application of zinc finger nucleases that we can also bring about the larger deletions in the genome. Yeah, so this is it for today's session. And now we'll meet in other sessions. So if you have any doubts, then you can ask. Otherwise, I'll be ending the session. Okay, thank you very much for being connected till now. So I'll end the session and we'll meet in the next session. So thank you.